Think about the last time you had a paper cut. Even though it was small, you look back at it a few days later and it's gone. One might ask how this happens, and I have the answer. Mitosis is a type of cell division, not to be mistaken with meiosis, which will be explained later. Mitosis is the form of asexual reproduction that multiplies body cells in your anatomy. Mitosis creates identical cells for each body cell. To create these exact copies, there is a cell cycle that must be followed. The first step of the cycle is interphase, which does three things in three separate processes. It generates cell growth and DNA replication and carries out cell functions. This is also the step that occurs first before meiosis and is conducted the same. The first step of interphase is named G1 or gap 1. Because the cell cycle is a cycle, it starts and ends with the cell dividing. The last step of the cycle is cytokinesis, which will be explained more in depth later. Cytokinesis is when the cells have finished dividing their chromosomes and the cell membrane along with it. Now, once this is done, G1 must occur. G1 is the development of new cells, which was divided in the last step. During this process, the cell will replicate organelles as well. If the cell is not actively dividing, it will immediately enter DNA synthesis, the next step of interphase. But if it is not dividing actively, it will enter G0, the resting state. Moving on, during synthesis, the cell pauses its normal functions. All that happens is DNA replication. This is done by unzipping strands of DNA. Other proteins, known as polymerase enzymes, begin creating new strands which pair with each half of the DNA. This occurs on every chromosome, making sure that everything is exact or a perfect copy. If the cell is somatic, it will enter mitosis and the sister chromatids will be separated. The next and final step of interphase is G2 or gap 2. To recap, the DNA has been replicated, so the cell now must add volume, specifically in the cytoplasm. In this phase, animal cells will replicate the mitochondria to produce enough energy for division. Plant cells will replicate the mitochondria and the chloroplast to provide sited cells and organelles capable of producing their energy. Once the cell is finally done preparing for division, the next step is mitosis. To start, the first step of mitosis is prophase. In prophase, the cell starts to break down some structures and build others up. This will set the stage for dividing chromosomes. In this process, the chromosomes will begin to condense, making them easier to pull apart later. The mitotic spindle will start to form from where the centrioles form. The mitotic spindle is made of microtubules, which are strong fibers that are part of the cell structure, like a skeletal system. The job of this step is to organize the chromosomes and move them around. The next step in prophase is the disappearance of the nucleus. Once this happens, it is a sign that the cell is now ready to break down. Next, of late prophase or prometaphase. Prometaphase, the mitotic spindle, capture and align the chromosomes in a line across the cell. Spindle fibers will extend to either side of the cell while this occurs. Continuing, the chromosomes become even more compact. As this happens, the nuclear envelope breaks down, releasing the chromosomes. Microtubules from earlier bind to the chromosomes at the kinetochores, a patch of protein found on the centromere on each sister chromatid. These are called kinetochore microtubules, and their function is to distribute the replicated genome from the mother cell to its daughters. Centrioles are at the center regions of the DNA. Chromatids are bonded the tightest. Next step of mitosis is metaphase. Metaphase begins when the mitotic spindle has captured all of the chromosomes, lined them perfectly in the middle of the cell. In the middle of the cell, now the metaphase plate. At this stage, the two kinetochores of each chromosome should now be attached to the microtubules from opposite poles. Before interphase can begin, cells will make sure that all the chromosomes are at the metaphase plate. Their kinetochores directly attached to the microtubules. This is called the spindle checkpoint. Clarifies that the sister chromatids will split in the next step. If they're not aligned, the cell will stop mitosis until they do. The step is called anaphase. This is when the sister chromatids pull apart from each other. The centromeres of the chromatids have broken apart and are now pulling to either side of the cell. The microtubules are not attached to the chromosomes, so now they begin to elongate and push apart, making the cell an oval shape. The process is driven by motor proteins, which are like machines that can walk across microtubules and carry their cargo. The cell is now almost done dividing. The next step that will occur is called telophase. This is when the cell begins to re-establish the normal structures on either side of the cell. The first thing that happens is the mitotic spindle breaks down. When this happens, the two nuclei form, one for each set of chromosomes. The chromosomes which were separated before now return to their stringy previous form. Finally, the last step of mitosis is called cytokinesis, which is when the cell division of the cytoplasm occurs. Finally, the last step of mitosis is called cytokinesis, which is the division of the cytoplasm. This will now form two new cells. Shortly after this step, the separate cells will each start to cycle over again, until they divide as well. Now in animal cells, the cell pinches itself inwards and creates a cleavage furrow. This is the pinch crease in the middle of the cell. Eventually, the cleavage furrow will be pinched so far inwards that the cell breaks, leaving you with two sets of cells, now ready to be in the G1 phase. Unlike this, plant cells, there is a very stiff cell wall that can't be pinched. Because of this, there is a cell plate that is formed in the middle of the cell, splitting it into two daughter cells. Now you have finished mitosis. Going back to the start of the video, I briefly explained meiosis. So how does this work? Meiosis is the replication of sperm and egg cells in the body. Before it begins, interphase must occur. When interphase happens in meiosis, the same steps take place as they do before mitosis. These steps are G1, synthesis, and G2. After this occurs, the cell is ready to begin the steps of meiosis 1. After interphase, prophase 1 occurs. This is when the differences from meiosis begin to appear. In mitosis, The chromosomes begin to condense, but in meiosis 1, they pair up, creating tetrads, or 2 sets of 2 chromosomes each. The chromosomes each align with the homologue partner so that the 2 match up at the corresponding positions to their full length. Synapsis also occurs during this phase. Synapsis is when the chromosomes condense. Once the chromosomes are matched up, one of each parent trades parts. This is called crossing over. When doing this, chromosomes are held along by a protein called synapsoidal complex. You can see the crossing over of the homologues under a microscope as schemata. After crossing over, the mitotic spindle begins to capture chromosomes and move them towards the center of the cell. This is the metaphase plate. This step is the start of metaphase 1. After they are attached in the center, the ends of the chromosomes connect to only one pole of the spindle. The homologues then bind to the microtubules from opposite poles. This helps them to align at the metaphase plate to prepare them for separation. Next comes anaphase 1, which is when the homologue chromosomes are pulled apart and moved to the alternate ends of the cell. The chromatids, however, remain and do not separate. The final step of meiosis is telophase 1. This is when the chromosomes arrive at opposite poles of the cell. In some organisms, the nuclear membrane reforms and the chromosomes decondense, but in other, there is another set of meiosis made specifically to separate the cells. The second set is called meiosis 2. This is only for haploid cells. These cells will move on to the next set without copying DNA. The first part of this set is prophase 2, which is when the nuclear envelope breaks down, if it hasn't already. The centromeres will also move apart. The first part of this process is prophase 2, which is when the nuclear envelope breaks down if it hasn't already. The centrosomes will also move apart, and the mitotic spindle forms between them. In metaphase 2, the chromosomes line up individually along the metaphase plate. In anaphase 2, the sister chromatids separate and are pulled together towards opposite poles of the cell. This is called segregation. Finally, in telophase 2, the nuclear membranes form around each set of chromosomes, and the chromosomes are decondensed. Cytokinesis splits the chromosomes into new sets of cells, eventually forming the final product of 4 haploid cells, which each chromosome has 1 chromatid.
Thank you, that's all.